This video will cover subtopic 2.6, the structure of DNA and RNA. So a list of understandings, applications, and skills that will be covered in this video. And just note, the skill we will cover in class. So we'll start with kind of the basics with the components of DNA and RNA. So DNA and RNA are basically two types of nucleic acids. And within cells, there are two types of nucleic acids that are present. So there's DNA and RNA. So DNA stands for deoxyribonucleic acid, and RNA is ribonucleic acid. And we'll talk about where those names come from when examining the structure of DNA and RNA. And so DNA and RNA are really polymers, so they're long molecules made up of smaller subunits. And these subunits are called nucleotides. So with DNA and RNA being polymers of nucleotides, so we can consider nucleotides being the building block of a nucleic acid. So a nucleotide is going to have three parts to it. There is going to be a pentose sugar, a phosphate group, and a base. And these three components are attached in a very specific manner. So the diagram here shows a simplified version of how these different components are attached together. So we have the pentose sugar, and the pentose sugar is a 5-carbon sugar. So think, in this case, ribose, or for DNA, deoxyribose. And the difference becomes between DNA and RNA comes down to the ribose molecule itself. So with DNA on carbon number 2, there is no hydroxyl group present on carbon number 2. So it is lacking in oxygen, so it is referred to as deoxyribose. On RNA, that hydroxyl group is present on carbon number 2. So it just gets referred to as ribose. So hence the name ribonucleic acid, or for DNA, where that hydroxyl group is missing, the deoxyribonucleic acid. Attached to carbon number 1 on the pentose sugar is the base. And the base is referred to as a nitrogenous base because it contains nitrogen. The base can have 1 or 2 rings in its structure. And on the next slide, we'll have a closer look at what those bases are. Attached to carbon number 5, the side group, is the phosphate group. The phosphate group is going to be negatively charged and acidic as well, or have acidic properties. So the bases, those nitrogenous bases found in DNA, can be categorized into 2 groups. So you have what's referred to as the purines, which have 2 rings in their structure, and then you have the pyrimidines, which have 1 ring in their structure. So looking at the purines, you have adenine and guanine, and for the pyrimidines, you have cytosine, thymine, and uracil. Just note that cytosine, adenine, and guanine are found in both DNA and RNA, whereas thymine is only found in DNA. The base uracil is found only in RNA. Those nucleotides join together through a process of polymerization. And when they join together to become those polymers, they are going to be joined by covalent bonds. So those covalent bonds will occur between carbon 3 on 1 nucleic acid and joining up, or carbon number 3 on 1 pentose sugar joining up with that phosphate group, which is attached to carbon number 5 on the adjacent ribose sugar. So the way when these are joined together now, this creates a sequence of bases for those nitrogenous bases. And the sequence of those nitrogenous bases is what makes up the genetic codes. So those nucleotides get linked together in a single strand by a condensation reaction, which everybody should be familiar with at this point. And as I mentioned before, those bonds are formed between the phosphate of one nucleotide and the pentose sugar of the next. So the phosphate group on carbon 5 joins with the hydroxyl group on carbon 3. And when you get all these nucleotides linked together, this creates what is referred to as a sugar phosphate backbone. As you have a carbohydrate and phosphate group that alternate, that create kind of the backbone of this nucleotide structure. So there are differences between DNA and RNA that you do need to know. So in RNA, the bases that are found would include adenine, guanine, cytosine, and uracil, which is only found in RNA. And then in DNA, it's adenine, guanine, cytosine, and thymine that is only found in DNA. As mentioned previously, in RNA, on the ribose sugar at carbon number 2, the presence of that hydroxyl group gives that structure the name ribose, so ribonucleic acid. In carbon, in DNA, on carbon 2, that hydroxyl group gets replaced with a hydrogen, and so is referred to as deoxyribose. And then in terms of number of strands, RNA is single-stranded, which we'll see a diagram of on the next slide, while DNA has 2 strands that are considered antiparallel, that are complementary and form a double helix. And so the next series of slides will go through discussing what those terms mean in reference to the structure of DNA. So this diagram just looks at the structural difference between DNA and RNA. So DNA is double-stranded, so it has 2 strands to it, where RNA is just a single strand of nucleotides. With the DNA structure, so looking more closely at that, so DNA does contain 2 strands of polynucleotides. With those 2 strands of polynucleotides, that sugar phosphate backbone is going to be on the outside, and then on the inside you will find those nitrogenous bases. So on the inside is where you'll find the A, the T, the C, and the G. And those 2 strands are going to be held together by hydrogen bonds between the bases. So it's hydrogen bonds that holds those 2 strands together. So in the following diagram, in one box that I've drawn here would be considered 1 strand of DNA, and then the other box would be considered the 2nd strand of DNA. So the blue part that I've just pointed to with the arrow is going to represent that sugar phosphate backbone, whereas in the other diagram, the red structure is the sugar phosphate backbone. The green is going to be the bases, and those dashed lines are going to be hydrogen bonds. And so it's hydrogen bonds that hold those 2 strands together. So with those DNA strands being linked by hydrogen bonds, it is going to be specific bases that have to line up on each strand to create those hydrogen bonds. And so in order to create those hydrogen bonds, adenine will always have to pair with thymine, and then cytosine will always have to pair with guanine. So it's the hydrogen bonding between those complementary pairs that hold those 2 strands together. And if you have a look at the diagram, what this means when thymine always pairs with adenine, for example, so if on one strand, at one location, you have adenine, on the opposing strand in a similar location, you will always have thymine. And then where cytosine is located on one strand, on the other strand in that same location, you will have a guanine. So adenine will always pair with thymine, or thymine will always pair with adenine. Cytosine will always pair with guanine, or guanine will always pair with cytosine. So these are referred to as complementary pairs. So they will always be found paired up on those opposite strands. So DNA now, with those two strands, are going to be made up of what's referred to as anti-parallel strands. So those DNA strands are anti-parallel to each other, which means that one strand runs from the 5' carbon to the 3' carbon, and the other strand runs from the 3' carbon to the 5' carbon. So the 5' end, or the 5' carbon, is the end closest to carbon 5 on the deoxyribose, and then the 3' end is the end closest to carbon 3 on the deoxyribose. So basically what that means is if we look at the one strand on the left, at the very end of the molecule, is going to be a free phosphate group. And so that phosphate group is attached to carbon number 5. At the other end of the molecule is going to be a free hydroxyl group on carbon number 3. And so that gets referred to as the 3' end. So one strand will start with the phosphate group at one end and will end with the hydroxyl group at the other end. The other strand now will run in the opposite direction, where the one strand ended with the free hydroxyl group, or the 3' end. The other strand now, the complementary strand, will now end with the 5' carbon, or end with that free phosphate group. So we have free phosphate group, free hydroxyl group at the end. And then that opposite strand now, or this strand here, which has the free phosphate at one end, will now have that free hydroxyl group at the other end. So those strands run in opposite directions to each other. Taken together, DNA will form what is referred to as a double helix. So those two anti-parallel complementary strands get wound together to form this double helix. And the way that the double helix is organized is that the sugar phosphate backbone is going to be on the outside, which helps protect those nitrogenous bases. Those bases are fairly reactive, so you don't want them exposed to the outside. You want them tucked inside in that structure. And then in this manner, this will allow those two strands to run in opposite directions so that they are anti-parallel. So the diagram off to the left shows that double helix structure of DNA, and there are regular repeating twists to that structure. So it twists on itself so that the bases are pointed towards the inside. That sugar phosphate backbone is found on the outside. So how is that double helix structure maintained? So it is hydrogen bonds that hold those two strands together, that hold those complementary base pairs together, but hydrogen bonds also will hold adjacent sections together as well. So hydrogen bonds between that sugar phosphate backbone will help hold it together. And that sugar phosphate backbone is also hydrophilic, so it gets positioned on the outside, so that'll interact favorably with that aqueous environment. And as I mentioned before, those nitrogenous bases are very reactive, so by them being pointed towards the inside of the double helix, they get protected. So with that elucidation of that DNA structure, it was quite a giant feat in biology. And it was two individuals that get credited with that, Watson and Crick. So to determine that possible structure of DNA, Watson and Crick actually used molecular models to test their ideas and determine if their model fit with the evidence that was made available to them. So they didn't perform any experiments themselves to figure out the structure of DNA. They took what evidence was available or what research people had done, and based on that research, they used models to build a structure of DNA that fit the evidence that they had. And so using that, they figured out the structure of DNA, but there were some failures at first. So the first model that they proposed was a triple helix, which is in this diagram here. So there's the yellow ribbon, and then there's two green ribbons wound around each other to create a triple helix. This was actually quickly rejected because they knew from Chargraph's experiments that the ratio of adenosine to thymine should be 1:1. And based on their triple helix, this ratio was not 1:1, so they knew it wasn't correct. And then this structure actually required too much magnesium to make it stable, much more than was actually available within the cell. So the second model that they came up with was a double helix. And building these models allowed them to easily visualize the relationship between the bases and allowed them to determine how the bases paired together and how they could form hydrogen bonds between each other. And it actually allowed them to kind of figure out this anti-parallel nature of the strands as well. Their model was also very handy because once they came up with this model of two strands that were anti-parallel, wound together in a double helix, it quickly suggested them a possible mechanism for how DNA gets replicated and for how genetic information gets coded. For their work in figuring out the structure of DNA, they were awarded a Nobel Prize a few years afterwards. Now, there was some controversy around this because the model they came up with was based on the experiments performed by other scientists. And most notably, one scientist by the name of Rosalind Franklin, who performed what's called X-ray diffraction experiments on DNA. Her work was actually central to them figuring out the structure of DNA. And so Rosalind Franklin, using this process of X-ray diffraction, generated these images of DNA, such as this image on the bottom right, which were very helpful in Watson and Crick coming up with their model. And in fact, there are some stories that say that Watson or Rosalind Franklin, didn't actually share her results or her experiments with Watson and Crick. Another scientist that she was working with actually took her work and allowed Watson and Crick to have a look at those results without her approval or her knowledge. But Rosalind Franklin unfortunately passed away before the Nobel Prize was given to Watson and Crick, and for a number of years she was not acknowledged as being like a principal contributor to figuring out the structure of DNA. She was later on awarded a Nobel Prize for her work, and is now recognized as a central figure in figuring out the structure of DNA. So in class we'll go through how to make a simple drawing for DNA structure, and we'll use some basic shapes to create our DNA structure. So we'll use circles to represent the phosphates, we'll use pentagons for the pentose sugar, we'll use rectangles for the bases, and then we'll talk about how all this gets attached together to create a DNA strand. So that is it for this video for subtopic 2.6.